Hi guys, this is part 2 of the nitrogen cycle videos. If you've not yet watched part 1, then please click on my nose and that'll take you back to part 1. Okay guys, this is where it starts to get interesting now. This is the denitrification process. So this doesn't usually happen in your aquarium, or if it does, it doesn't happen at a very high level. And this is the conversion of nitrate into either nitrogen gas or nitrous oxide. Nitrous oxide is also known as laughing gas. It's what you get at the dentist. So this is a process mediated again by bacteria. These bacteria are known as facultative aerobes. That means ideally they like to use oxygen. We use oxygen. Most of your common bacteria will use oxygen. Oxygen is used as a final electron acceptor in the electron transport chain. If you want to know more about that, again, leave a comment and I'll get a fact sheet out to you that details all this kind of thing. Sorry, ignore my aquariums, they're making a noise in the background there. So, like I said, the facultative aerobes, which means they would prefer to use oxygen. However, in this case, they can be forced to use nitrate as the final electron acceptor instead of oxygen. This means that you can't have a high oxygen environment in your aquarium for this to happen, which you couldn't achieve in the water column because you've got fish living in there that need oxygen. So the way you do this is in a marine system you would use something like a deep sand bed. In a regular freshwater aquarium you could use a deep substrate layer. It's why it's not recommended to use things like play sand that can compact and create anaerobic areas, because it's quite a complicated process, and other things can be produced like hydrogen sulfide, which when bubbled through water makes sulfuric acid. So there are other byproducts of this process that aren't desirable to have in your aquarium. Quite popular now are the nitrate removal things you can get. You can actually do this by, if you get some airline and coil it round a piece of PVC piping loads and loads and loads of times and have a slow drip coming through there, run your water through there at a very slow drip, you'll get a biofilm coating the inside of that tube, and the oxygen in that water will be used up before it comes out of the tube, and you'll get a build-up of these bacteria that can use nitrates and convert them into nitrogen gas or nitrous oxide. So that's one way to perform it in your aquarium. Another way is through, if you have highly porous rock. So I know Pond Guru, a lot of you guys will be subscribed to him, he's got quite a lot of followers. He's always talking about this biohome media that he's got. It's very porous, but those fine pores allow for bacteria to coat and use up that oxygen in the water that's in there, and by the time sort of that water's got to the core, there's very little oxygen in it, so the bacteria there can utilise nitrate and other compounds in anaerobic conditions. So that's a little bit about the process. Let's talk about some of the species, because some of these are really interesting. Again, this is more a microbiology thing now rather than a fish tank thing, but I think it's quite interesting. So there's Thiobacillus, which are known to do this. The only problem with those is there's something called a thermophile, which means they prefer hot temperatures, anything above 50 degrees, which rules them out of the fish tank, because, you know, what are you going to keep at 50 degrees? There are actually some fish that are found at very high temperatures that we don't really keep in the hobby. But if you have a look on the website Seriously Fish, there's some really cool fish that live in all kinds of crazy environments, right down to like pH 3 and temperatures above 40 degrees. It's really interesting. Anyway, back to bacteria. The most common one you'll find in your tank, and it will be present in your tank, these bacteria will either work aerobically, so they won't be doing this, or they'll be at such a low level because there's not the conditions aren't there for them to utilise nitrates. You won't really notice it. So things like Pseudomonas denitrificans you'll probably have that in your aquarium. Pseudomonas are quite a large group of bacteria. Ones that you'll know are Pseudomonas aeruginosa. That's a common respiratory tract pathogen. It can be very nasty. It's very nasty for people with cystic fibrosis. So the Pseudomonas species are common in water. They're a waterborne gram-negative organism. The one that's really interesting, that I think is really interesting, is Paracoccus denitrificans. This bacteria has huge potential. If anyone like CChem is watching this, this is a bacteria that can produce nitrous oxide aerobically. So it doesn't need these anaerobic conditions to do this. It's found in wastewater, it's found all over the place. It's a very interesting bacteria. So, like I said, this process needs to take place in the absence of oxygen. However, not with this bacteria. Also with this bacteria, it can utilize heterotrophic nitrification, which means it can do both stages of the nitrification process that we talked about in the first part of this video, and it can also it's also responsible for denitrification. So this can take ammonia and take it right through to nitrous oxide or nitrogen in the presence of oxygen. So this is one bacteria to do everything. If you could do some research on this as something like CChem or other aquarium products manufacturer and sell this bacteria, you've got something quite cool there. So there's something to think about. That's pretty much it for the video. If there's anything you feel I've not covered, leave a comment below. I'll send you out the little sheet with all the metabolisms on and a little bit more explanation, see what you think about that. It's one that's been knocking about on the internet for a while. I've uploaded it to various websites that I've been part of before. It's basically it's a simplified version of Dr. Timothy Hovenick's research. And I've passed it by him, he's okay with it, so there we go. So the takeaway message from this video is the importance of your filter. Your filter is a biofilm of bacteria. It's a community of bacteria that can take in ammonia and pump out nitrate. In your filter, that's all you're going to achieve really. With certain medias, you can then take that one further, try and remove nitrate from there. Usually you would remove nitrate through water changes, through heavily planting your aquarium. Floating plants are quite good for this as well. And plants use nitrates in a whole different process, producing amino acids and proteins, and then as they break down, they release them to the water and the cycle continues. So you can see why it's called a cycle. The things to take away from this are that's the key thing. Remember, temperature is important, pH is important, light is also quite important. UV light is actually quite harmful to most bacterial species, so your filter kind of wants to be in a dark place, which is why external canister filters are very good. They're completely pitch black. So I think of your filter as your life support system for your tank. A lot of people say that, and it's true. Other things that will help with this process are things like porous rock. So I have a lot of volcanic rock in my tanks, and that helps this process, particularly the denitrification process. It's one of the reasons you'll see algae sometimes building up on things like volcanic rock, because you've got an abundance of nitrates being formed at the rock. So there you go, guys. That's my video on the nitrogen cycle. Hopefully this has helped. I know it's been a long video. Thanks for watching till the end if you have, and hopefully I've done something cool with little graphics here and whatever, but we'll see. So I will catch you next time, guys. Thanks for watching, and come back next time. Thank you.
Thank you all for watching, and I'll see you next time. Bye.